Okay, we are going to get started. We got a lot of people already have joined. So my name is Gloria Isabel Giraldo Calderon, and I will be one of your instructors today. Please let me know, are you able to see my slides? Somebody from the outreach team or somebody in the chat?
Yes, we
can see them.
Thank you. Okay, so before we get started, let me introduce you to Dr. Marianne McDowell from the University of Notre Dame, who is one of the 3 co-PIs of the ViewPath database project. Thanks for being with us today, Marianne.
Thank you, Gloria, and welcome, VectorBasers. We're super happy to have you and excited to show you the new ViewPath DB VectorBase resource. As Gloria mentioned, I'm one of the joint PIs of ViewPath DB. And some of you may already know that I took over as the PI from Frank Collins in the previous iteration of VectorBase. Before we get started, I'd like to give you a little history about the Bioinformatics Resource Center, which is funded by, or the program, which is funded by the National Institute of Allergy and Infectious Disease within the National Institutes of Health. And that's what funds these programs like VectorBase. The program started in 2004 when they awarded 8 different VRC contracts. And the funding cycles go in 5-year increments. And after in the second iteration, they actually consolidated to 5, or actually really 4 VRCs, 1 for viruses, 1 for bacteria, 1 for eukaryotic pathogens and fungi, and then of course for vectors or VectorBase. In the most recent iteration of the funding cycle, the NIH consolidated even further and only awarded 2, 1 for viruses and bacteria, and the other for eukaryotic pathogens and fungi and vectors. And so now we're calling this ViewPath DB. And so it really, the program forced us in a way to merge, but we were really excited to do that and bring together the best tools in both the ViewPath DB and the VectorBase resources. So we started the merging process in September of 2019, and we're almost complete. Everything will be done in May, and at that time we will be removing the legacy VectorBase site. So we're really happy that you're undergoing this training, because pretty soon this will be the only one that you can use. As I mentioned, we leveraged the strengths of both VectorBase and ViewPath DB. The tools and resources, but also we merged the 2 staff and outreach personnel together. So I think that this is really going to result in improved service to the vector community. So I know that change can be difficult, and some of you have found the new resource a little difficult to get started with. So we really appreciate your patience and your willingness to attend this training. And as always, we'd love to have your feedback and how we can improve the resource. I'm confident that the outreach team will be able to get you over the hump. There were a couple of things that I'd like to mention. So many of you already know that VectorBase and now ViewPath DB collaborate with EBI. And some users have found it difficult to change from the Ensemble Genome Browser to the JBrowse browser. So there are many reasons we chose JBrowse, but one of them is that the Ensemble browser is scheduled to undergo a major rework. So it was going to change anyway. We just didn't really know the timing of that. In addition, some legacy VectorBase researchers really liked the BioMart capability in Ensemble. That too is scheduled to be replaced. And so we came up with some different functionality to help you do the same tool. So the ViewPath DB staff is extremely dedicated to making the best resource possible. So I'll reiterate, please do not hesitate to reach out to us. And I hope you enjoy today's webinar, and I look forward to working with you all in the future. So I'll turn it back to Gloria. Thank you.
Thank you, Marianne. Thank you very much. Okay, now some logistics. Okay, this webinar will last for 1 hour. This is the outline that we have for today. But the main goal here is to answer your questions, and that's why we are doing this live. So our topics are the basics of how to use the VectorBase website, and are also addressing some of the more frequent questions that we receive from you. And how are you able to send those questions here in the website? You can click in contact us and send us an email, use the links to Twitter and Facebook to send us a message via social media, or here in the community chat of the website. So not only during this webinar, but always the outreach team is ready to answer your questions and hear your feedback, using all these communication channels. This here, I'm back here with the slides. Okay, so this here is an image of how the webinar is looking to you. So at any time during the webinar, please type a question here. And in Omar, Sam, Suzanne, and other members of the outreach team will answer the questions you post. We are recording this webinar, and the video will be up in our YouTube channel. And again, the link from here, from the top of the page. And if you want to watch the previous webinars, you can come to the homepage, click on Help, learn how to use ViewPath, click on Webinars, and you will find links from the previous recordings, or you will find links for the upcoming webinars, and of course, later today or tomorrow, we'll upload this very same video. I also have here, as Marianne said, other pages from ViewPath. So as describing the invitation we sent out, this is an introductory webinar series, and we will use vector-based examples only, but the layout of all the ViewPath sites is the same. You can do anything we're going today in the demo with any of the other ViewPath sites, from fungi or parasites. And here I'm showing you plasmodbs, and here I'm showing you trip database. Okay, so today, the instructors. So we have Omar Harp from the University of Pennsylvania, who will talk to you about site search, Genome Browser, and Galaxy. We have Suzanne Warrenfeldt from the University of Georgia, who will talk to you about Multiblast. We have Sam Rand from the University of Notre Dame, who will talk to you about MapView. And myself, Gloria, I'm based at the University of Notre Dame too, and I will present the homepage and give you a general overview of how the website is organized, where and how to find genomes and other data sets. I will show you how to construct your very first strategy, which is the system that replaces Biomart that Marianne was talking about. And advanced details about searches, complex strategies, and nested strategies will actually be the topic of our next webinar, which is in 3 weeks. And I'm also going to talk to you about the gene pages. Okay, so now let's officially get started. Something very important is that you have the possibility to have your own account in the website. And if you click here, you can, in your name, if you already have an account, you provide your details, and you can save your search strategy or your blast jobs in your account. But optionally, you can opt in to receive release notes. And for example, here I have only checked VectorBase and ViewPath, but you can check as many as you want or none of them. Because you can also see all the release notes that we sent going from to the home page. Sorry, it's the go to webinar in the middle. Click on news, and here you click on the release notes. The body of that email will have something like this when we present all the things that are new. But of course, if you're a social media person, we are also going to post that there. And in social media, we'll also share scientific papers or other relevant information that we think will be useful for you. Okay, about the website now. So here we have these 8 navigation bars. And today we are going to focus on 2 tools. And from this list here, we are going to demo a few of this. And I'm going to show you right now data. In data, I want you to look at data sets in VectorBase. So if I click there, data sets in VectorBase, I get loaded this page. So notice also that we are in release 51, and this came 16th of March. And now for this release, we have almost 500 data sets in the website. You can use this box here to filter them. So if I start typing genus, for example, Edis, notice how out of the almost 500 data sets, 141 are for Aedes aegypti. And you can do a more specific filter, clicking on this down arrowhead and selecting only the organism or the category. You only want to look at RNA-seq data. And here in category is only showing you RNA-seq. So it's telling you that we have 327 RNA-seq data sets from all the data sets in VectorBase. So we have both data sets from external databases such as KEGG or chemicals from EBI, and also data sets from scientific papers, which could be, for example, as I said, from an RNA-seq transcriptome. So we do not generate the data. Scientists like you or external databases generate the data. VectorBase is just the home of this data. But more about how to use these data sets in the search strategy, which will come next. Okay, so all these data sets are first deposited in one of the INSDC archival repositories, most likely NCBI or EMBL-EBI. And again, to reiterate what Marianne just told you, this is why when you notice some supercontig, scaffold, or chromosome name change, it's not something that we do. The authors of the data submit to INSDC, and from there we copy to VectorBase. So please, if you notice a name change in any data source and need some help mapping the old names, we can help you with that. Just let us know. Okay, so in this page that we have all the data sets in VectorBase, there is a subset that includes the genomes. And for the genomes, we have created a special page, and you can access from here, organisms. If you click there, you will get loaded this page. So this is telling you that in VectorBase, we have 46 organisms or 46 genomes. And some species have more than one assembly. For example, here, Aedes albopictus has 2. What is one of the most important pieces of information in this table? This column right here, because it's telling you what is the assembly version and the gene set version. So for example, for Aedes aegypti, and for all the species, is the first letter of the genus, the first 3 letters of the species, the first letter of the strain, the version of the assembly, in this case number 5, and the version of the gene set, 0.3. And this is important for your personal records, for your lab notebook. You need to put that in your poster, in your thesis, on your paper. Why? Because this will allow you to correctly describe your methods, and other scientists will be able to reproduce your findings or know why they are finding different things. And with that, Omar, you can take it away with site search, and I will come back later.
Okay, thank you. Let me make myself the presenter and show my screen. So I think everybody can see VectorBase. So welcome everybody. And again, as a reminder, feel free to add your questions in the question panel as Gloria indicated. So site search is a tool available to you at the top of all pages on VectorBase. And this allows you to enter a text word and search for any word of interest. So anything you're interested in looking for on the site, you can type it in site search and then return results. So for this demo, I'm just going to type the word multidrug. And so obviously you can select anything, kinase, phosphatase, or anything you're interested in. And once I run this, it's going to return any record in the database that includes the word multidrug somewhere in the record. And you'll see here, I returned 1,168 records that have the word multidrug in them. And they're listed at the bottom here, but on the left-hand panel is a very nice filtering mechanism that allows you to filter on the different record types. And also it gives you a quick summary of where the results were found. So at the top section here, I can see that there are 1,150 genes that have the word multi-drug somewhere associated with them. And then there's also 18 compounds that have the word multi-drug associated with them. And then as you scroll down, you will notice you can see a filter by organisms, and it shows you the number of hits in the different organisms. If I expand all, for example, you'll see the distribution of the hits across all the organisms within VectorBase. So this is a nice and quick way for you to get a quick sort of 10,000-mile view of where you're finding the results. You can use this panel on the left to filter the results that show up here in the central portion of the page. And there are different ways of doing this. For example, if I'm interested in only finding results that are associated with anopheles, for example, I can just start typing again. And as you type, it starts filtering the results here. And you may be satisfied that, oh, I'm finding what I want, or you may have to be more specific. So for example, I can maybe use the species instead of the genus. And now I can go to, I see Anopheles gambiae right here. I can select this. There are 20 results. And if I click on apply, this will filter my results in the center here and show me only the 20 results that were associated with Anopheles gambiae. And you'll notice that they're all actually gene results right here. And I could further filter this by saying, well, I only want to see the gene results. So I can click on gene. And you'll notice now that, again, I'm just viewing the genes here in the central portion. But now you can filter additional fields within the gene results themselves. And these results were found in orthologs of your genes of interest, the hits, and also in PDB, so the protein data bank for structural protein structures. And so these are similarities to PDB that had something to do with multi-drug. And I can obviously, at any moment, I can clear all my filters and return back to my initial results if you're happy with that. You can obviously scroll through these. One nice thing about the central portion here is that you will notice that there's the gene that was found. It tells you, what is this gene called? In this case, it's an unspecified product. It tells you the organism, and then it shows you where the search was found. If you expand this, it tells me that a multi-drug was found in the orthologs, and it gives you, in fact, the context of where that was found. So that's really helpful for you to determine whether this is something actually a real hit or something you may be interested or not interested in. And then the last thing I will point out here, well, actually two additional things. One thing you'll notice here, there's this hide zero counts. So we're only showing you where filter items or fields that were actually had a hit in them. If I uncheck this, you will notice now it shows you all the other fields that are available that did not have a hit in them, right? And so obviously not useful when you have a bunch of zeros, but it tells you all about other things in the database that you could find results in. So depending on the search you run, these numbers will change, and this hide zero obviously will give you different results, obviously depending on where the word was found. And the last thing I will show you is that if you filter on any of a specific record like genes or compounds, I filtered in this case on genes, I can, and if you want to work with this list of genes elsewhere on the website, you can further take this and export your results. So this little button here up on the right-hand side allows you to export the results that you found here into a search strategy, which Gloria is going to demo to you anyway in a little bit. But this allows you then to extend the results, so the 1,150 genes that had the word multi-drug in them, and do more work on them and understand them better. So I will stop here. Maybe actually before I switch it over, you will notice in the site search, once you run a search, there's a little arrow here that shows up. And this allows you to quickly, instead of having to type multi-drug again, let's say I want to go back and look at the results, you obviously can click on your back arrow in your browser, or you can click on this little arrow right here, and this will take me right back to the search results, and I can start over with other filtering and then bring it back to the search strategy. All right, I will stop here and turn it back to you, Gloria.
Thank you, Omar. Okay, I'm going, can you please stop sharing so I can share my screen? Marianne also mentioned this, and this is for former Biomart users. So I'm going to translate to you everything that you were able to do with Biomart, how you can do it with the new search strategy system that, well, new for VectorBase, all users, but how you can translate the Biomart usage to the strategy system. Okay, so when you come to the homepage, and I'm also going to show you this live, you will start you will select where do you want to start. Two important points to start. You can have a custom list of genes of interest. So you will start from gene ID, or you can start with the entire genome as your gene list. So you will start from organism in the taxonomy category. And here, please be very careful because there is 3 places in which organisms are located, but you want the one that is under the taxonomy category. Okay, from there, this is the screen that Omar just showed you. You will have your strategies and you will start growing them one step at a time. One very frequent use that people had with Biomart was to convert their list of orthologs, their list of genes to orthologs. So you will click an add step and it will give you the option to transform your genes to orthologs. You can analyze your results. Click here to perform an enrichment analysis, and that could be gene ontology, for example, or a metabolic pathway enrichment. You can download your results and you have different options. So for example, if you click here, there are 1, 2, 3, 4, 5 options. The top one, it gives you, you can download a table with gene metadata. But if you select the third option, again, coming from here, from download, you can obtain, for example, the sequences. You can visualize also when you are looking the quick view here of the data. You can add or delete columns, and you click here, and these are all the options that you have. Okay, so now let me show you that in the website. Okay, here in the homepage. Let me move this around a little bit. Okay, so in the homepage, you start here again, and as I said, you can type gene ID, for example, and you click here to start your search, or as I said, organism. And notice it comes in 3 different places, but if you want all the genes from a species, you want to select organism. Okay, so let's go back to the initial one that I had in mind, gene ID. I select this search. And here, you can start customizing it. So I'm going to select, I have my list of genes already instead of this one. And I can also, upload a text file. Click on get answer. And I have here my 12 genes. If I notice that this is a step 1, and if I hover with my mouse, I get this edit button. It's reminding me what I did, and maybe I made a mistake, so I can revise my strategy. Maybe those are not the genes that I want, or I can change anything. And from here, I can add a step. So I'm going to show you the one that I just did. I want to transform this to the orthologs, and I want this in Aedes aegypti. So I can type Aedes, or I can type a part of the strain, LBP, and you select the species that you want, and you run the step. So notice how I'm transforming this list of genes, and you can name this strategy. Click here on the pen. This is a demo strategy for webinar, or anything you want to call it, and you save it. Here you have other action items. For example, you can copy the strategy to make some variations of it. And you can have this to remind yourself. So in step 1, I upload a list of genes. In step 2, I convert to edits orthologs, or anything you want to do. You can save the strategy. I want to save this, and notice that I had an asterisk before and now it disappeared because I have saved it. And now that you save it, and notice I have my account, I can share this with anybody I want. You can even publish this in your paper. So have this as an image in your article, and also share the link of it. You can delete the strategy or just not show it here with your other strategies. For example, I did the same strategy earlier. This is not going to delete it from all my strategies. It's just deleted from my view here right now. And to help you get started with the strategies, we have created 13 public strategies for you. So if you want to get started, you just click on any of these and we'll open them in the panel here. And I have here custom list of genes of interest, entire genome as gene list. These are the two that I just showed you in that slide of how to get from biomarker to the strategy system, but many other strategies that you can copy and modify and make your own. And if you have a research question and you think the strategy system can help you, but you don't know how, send us a message and we will be more than happy to help you construct your strategy. Something else that I want to show you, are you able to see my PowerPoint slides, somebody?
Yes.
Okay. So here what I want to show you is what does it mean for you that VectorBase has almost 500 data sets. It means that you have only to learn how to use VectorBase and have access to all these different data sets from many different databases. It means that, for example, if I publish a paper on mosquito immune genes, I'm only analyzing a low percentage of the total protein coding genes, and all the other data is there to do new hypothesis-driven research. And even people can take a look again at the same immune genes, but from another perspective and create a different hypothesis. So in summary, it means that you can run in silico experiments with real in vivo data at no extra cost to you. And here, this is one data set that we have currently available in VectorBase. And so I'm going to quickly tell you what this is about. So this is the life cycle of a mosquito, and they had females that they give sugar, females that were mated, and they had blood meal 48 hours, and females 96-hour plus blood meal, and which were ready to lay some eggs. And from both the males and the females, they had different tissues. So they collected 10 different tissues for females and 7 different tissues for males. And again, this data set is in VectorBase. And you can, what they did in the paper is that they compare these different tissues in males and females, and they also compare the 3 different states of the females for each tissue. So what did they provide us? With 28 different samples for males and females, the different tissues. So this is a tissue-specific catalog of genes for different adult stages, and they are comparing males versus females or females versus females. And we have the raw data because the authors deposit this in SRA, and they focus on the neural genes, and they focus this as a neurotranscriptome, but in fact they have the whole set of genes, and they analyze these when EDS-GPT was in version 3 of the assembly. And remember, I just told you we are now in assembly number 5. So what this means to you is that because this data set is in VectorBase, you can ask different questions using the same raw data. So remember, we are now in this assembly, and we internally run the pipeline to have these data updated with the new assembly. And actually in the paper they mentioned that they had some missing genes, or they were absent compared to other species, or they have found in the transcriptome that they were not in the genome at that time. So you can be able to find those genes. what are the type of questions you can ask with our strategy system using this data set? Mating-induced changes in males versus female, and you can take the brain, the genitalia, or the ovaries from this study. You can compare sugar meal versus blood meal with the 3 different status of the females, or because the females had been given blood, you can study egg development or egg laying behaviors. You can also study hearing, vision, and chemoreception because they collected antenna, brain, legs, proboscis. But this is just an example of what you can do with this data set. But remember, we have many more. You can select specific genes or families or group of genes or only hormones, whatever you are interested. So I will really encourage you to look at the strategy system. And now we are going to completely shift gears here, and we are going to talk about population biology data. And now Sam, it's all yours.
Thank you, Gloria.
Okay.
Yes, so I'll be just demonstrating our map view interface today. And for previous vector-based users, you may have known this as Pop Biomap. And it's for visualizing field collected data that is spatially and temporally resolved. And I'll just be showing the features rather than a deep how-to, but please do reach out if you're interested in using or contributing data of these kinds. And we have a number of different views shown here, and they're customized slightly different for the different kinds of data that we have. So right now I'm showing the IR genotype view, and here I have filtered by Anopheles gambiae, and you can add other filter terms, date searches, seasonal searches, filters, etc. here by using the autocomplete search feature here, or you can use the facet legend window over in this bottom corner. So I'll change from right now it's faceted on allele to faceting by locus, and I'm just going to add the KDR L1014 locus. And so that filters just by data which we have on this. And I'll switch it back to allele so you can just see the allelic breakdown at that locus. And I'm going to click to zoom in, and as you can see, the data disaggregates, becomes more spatially resolved as I click in. I'll zoom back out. You can undo that by zooming back out, and then the marker points will re-aggregate. And as you can see, you can click on a point and see the allelic breakdown that we have reported at that point. Here I'm looking at a different data type. This is mosquito population abundance surveillance data, and much of the data that we have comes from mosquito abatement districts in the US, but we are in particular trying to acquire more African population dynamic data sets, so please do let us know if this is of interest to you. And here I will do the opposite. I will actually remove a filter so you can see more data, and you can see that we can change the temporal resolution and do other kinds of graphical things to better drill into the data and explore it. For insecticide resistant phenotypes, which I show here, we try and curate as raw data as we can so that then you can go and do further explorations. So for example, here I'll add the deltametrin filter. I'll zoom in.
Quickly, Sam, if you can show people how to get to map view. I think you skipped that point in the beginning or went over it very quickly. So there are a couple of questions about that.
Yes, I will do that at the end, yes.
Perfect, okay.
So here we clicked on a data point, and you can see all the results of the underlying assays and how we got this data. And indeed, so this is, I showed you the graph view, but we also show data in tabular view. You can download all the raw data as a CSV file. And even if you have this interesting view that you want to share your colleagues, you can click download or the share button, and that will create a link that you can email to your colleagues so they'll get what you're seeing on your screen. And to answer Omar's question, which I am sorry I should have started with, from our homepage, you can click on Tools, map view to access the map. So the two final views I'll show just very briefly. We also have data on pathogen status. So as you can see here, this shows the change in positive test results over time, and it shows both the number of tests that were performed as well as the positivity. And we have our blood meal view. So for example, here using analysis of the host blood meal sources, you can get an idea of the zoophilia or the anthophilia of the vectors that we collected. And again, we are continuing to add more data. Most of the data that we acquire comes from paper curation, although we do work directly with data providers. And we also work for insecticide resistance data. We work closely with IR Mapper, if you're familiar with them, to ensure that our data is synchronized and that we share the curatorial burden. So please reach out if you have any other questions. Thank you.
Thank you, Sam. And now we are going to Suzanne to learn about multi-blast.
Okay, can you see me and hear me?
Yes.
Yes.
Okay, good. So I'll define BLAST in hopefully 30 seconds. So BLAST stands for Basic Local Alignment Search Tool, and it's a program that finds regions of local similarity between sequences. The program compares nucleotide or protein sequences to a sequence database and then calculates the statistical significance of the matches. We link out and we use NCBI BLAST as our algorithm. So anything initiated from VectorBase will use that algorithm. And they have a fantastic chapter there about BLAST that they've written. So I suggest if you need to learn more that you go there. Okay, so BLAST Beta is our new tool in the new VectorBase that is multi-BLAST capable. You can query up to 100 sequences at once with this tool. It's available from this tools menu under BLAST Beta, and it'll eventually replace our old tool, which only does one sequence at a time. So I'm going to click on that, and I'll go to the tool, and what I see, I can move this, is that I have, 5 parameters here that I need to define and before I can initiate my BLAST. So these parameters define the database that we'll BLAST against, the BLAST program that we'll use, and then the query sequences that we want to find similarity to. For this example, I'm interested in proteins. So I choose that and automatically grays out some BLAST programs that are unavailable. I have a choice between 2, BLAST P. If I want to learn more about what those are, I can use this help bit right here where they're defined, and then I also encourage you to go to the NCBI BLAST page to learn more. So I'll use BLAST P. I can BLAST against all of the organisms that are in VectorBase, but in the interest of time, I'm just going to use BLAST against 80s organisms. And now I want to input my sequences that I want to look for similarity from, and you can actually enter them as a file, choose a file with this option here from your computer, but I'm just going to go ahead and cut and paste. And your sequences need to be in FASTA format, which is, a def line that ends, that begins with a little right arrow, I guess, followed by the sequence. And the def line will become the name of the sequence in your BLAST results. So you want to make, you could make this descriptive if you like. And then I need to give my job a name. And you have the option to change these advanced BLAST P parameters. Now we have chosen all of these parameter values that that commonly people use in their blasts, right? This is a pretty average blast initiation that we're going to do. I'm just going to leave these as default. If I want to learn more, if I know what I'm looking for in terms of a score matrix, I can change these parameters, but I'm not going to do that right now. I'm going to go ahead and initiate my blast. So as you see, the blast ran pretty quickly. I was only taking 4 sequences against a couple of different proteomes, and I'm taken to my blast result, which has a summary at the top and then 2 tabbed pages below. The summary, reminds me of what my the database that I set up and the name of the job that I ran. The combined results show all of the hits for all of the sequences that I blasted. We can see there are 108 genes returned, and these genes, these are genes whose protein products showed sequence similarity to one of my query sequences. And so the first one, the gene returned is short chain type dehydrogenase. And in this table here, we list some of the statistics that came back with the blast. You can download these results in a myriad of formats. You also have an individual results tab. This is where you can view detailed results for each of your query sequences. So I can toggle between my sequences here. I'll choose my 3rd sequence, and this was the name. This is the def line actually from the FASTA file that I submitted. So, and then these 3 tabs then are populated with the results of my 3rd sequence blast. So the first tabbed page is the blast results. And this is what comes back from NCBI blast. It has a list of the sequences that your sequence hit, and then statistics that it's calculated. And if you scroll on down, you can look at the alignments. The second page then is a more familiar VectorBase now result with the gene ID in the first column and then associated data in columns next to that. I can go ahead and add any data within VectorBase that I want to. And for that, and then we also have a 3rd page, a genome view that maps the hits for this blast back to the chromosomes and genome sequences to which they belong. Looks like that is taking a while to load. I'll go back to my gene results, and I can download this in a bunch of formats. I can also export this to the strategy system where, like Gloria said, I can add a step and intersect these genes or transform them to other organisms. Now, here I am in the strategy system, and I want to get back to my blast results. I go to my workspace and my blast jobs, where I'm taken back to any of my jobs page where all of my jobs are saved. If I'm a registered user and I'm logged in, then my jobs are saved. And from here, I can also go back and initiate a new job. I think that's it.
Thank you very much, Suzanne. Now I become the presenter again, and I'm going to talk to you about the gene pages. But before I do that, I told you before that I was going to show you some slides, and I was going to show you live how to do things, and I think I completely forgot. So just a little bit more about the strategy system that here. So if you come to the home page, remember I tell you, you can type gene ID, or you can type organism, but if you want to use the RNA-seq dataset that I just demo, or any of the other, data that we have. You type that RNA-seq, and you click on that RNA-seq evidence, and when you click there, all the RNA-seq data sets from VectorBase are going to load, and you just type a keyword to select the one that you are interested in. So for example, I'm interested in this neurotranscriptome that we just took a look, and there is different options of how you can query this data. But again, remember, we are going to look at the details of the strategy system next time in 3 weeks. But I just wanted to let you know how you can get to what I show you in the slides. And once you select any of these options, for example, if I select the fold change option, you will be able to select between your reference samples. Oh, I want to compare the female 96 hours against, I don't know, female 48. I'm making something up here. But make a sensible question, knowing about the biology of the organism you are working with. And with here, this graph is going to help you construct a strategy, get answered, and you get again to the strategy system, but with more details about this in 3 weeks. Okay, now to the gene pages. So when you are in the strategy system on any part of the website, if you click on a gene ID, you are going to land in a gene page. You can also use site search and type the name of a gene, and you can get here from click on your results and get to the gene page. Or you can just in the site search type a gene ID. When you do a gene ID, it's a very specific query, and you will get this card highlighting, okay, this is most likely the hit you are interested in. So if you click there, the gene page will load, and this is the table of contents for this gene. So here we have the gene ID, the name or description, where the location, the species to which it belongs. And for example, here I can go, if I select the different places in the table of contents, I can navigate, or I can type a keyword. I want to get to the BLAST-P and select that. I can go back to the top of the page, or for example, I can go to Apollo to annotate a gene, but you can also get from to Apollo from the tools menu, or you can go to the Genome Browser. So from here we have little windows in different sections from which you can look at the Genome Browser, but you can also go directly to the tool. And with that, I will give it up to Omar, who will talk about the Genome Browser.
Okay, thanks, Gloria. So once again, just as a reminder, the tools menu is really useful. Actually, all of these little menus on the top are very useful. They get you to different places on the website, including the tools menu, which includes many of the things that we talked about already, map view, blast, but there are many additional things that are quite useful. I'm going to highlight 2 additional tools that could be useful to you. One of them is a Genome Browser. And for that, I'm just going to go to the tools menu and go to the Genome Browser. That said, you can access the Genome Browser from many different locations, including the gene page that Gloria was just showing you. There are links to the Genome Browsers from different portions of the gene page, where it'll take you to that to the Genome Browser with that section already loaded. So if you're looking, for example, at an RNA sequence data set on the gene page for your gene of interest, and now you want to look at the coverage plots for that particular RNA sequence experiment, you can click on the link from that experiment on the gene page and get directly to that gene in the Genome Browser with the appropriate tracks loaded. So if I go from the tools menu, it's just going to take me to a generic point in the Genome Browser. I'll give it a second to load. There we go. And you'll notice here I had a bunch of things I was playing around with last night, so I have a bunch of tracks turned on, but depending on, it remembers what you did last time, and so, which is nice. And so if you end up doing a bunch of work and then you go away like I did, and you come back, next day, your previous tracks will be turned on. So a couple of key features. So again, we have recorded webinars that go in through the Genome Browser in great detail. Also, the Genome Browser we use, JBrowse, is developed by the Gmod Consortium. They also have a number of tutorials that describe basic JBrowse functionality, and I definitely recommend that. And as usual, click on the Contact Us link if you are trying to do something in the Genome Browser and you're just not sure how to do it. We'll be more than happy to help you. You can quickly switch between genomes. I think by default, this is it's an 80s genome, but I can go in here, for example, and I can switch to any of the genomes that are in VectorBase. And so, for example, I can pick Anopheles gambiae here. It'll load now a region in, and this is, again, it's just a predefined region in the Genome Browser. Again, it remembered my previous tracks that I had turned on. And I'll show you quickly how you can turn on tracks. I'll show you a couple of features that are important in the Genome Browser, but it's full of many additional features. So if there's something you think you should be able to do in a Genome Browser and you can't find it, just let us know. You can move around here by clicking a specific ID, or you can select a different chromosome, for example, or contig for that matter. So that's a quick way to navigate. Another thing you can do is you can zoom in and out in a particular region. So I can zoom out and it'll go further out, or I can zoom in. I can click on these little magnifying glasses, or I can click and drag within the window, and that will allow me to then select a specific region. So when you click and drag, it will give me the zoom region, and now I can zoom in specifically to this region, and it'll load all the data in there. Obviously, the more tracks you have turned on, the more time it takes to load. It's just a matter of how much information is being transmitted back and forth between VectorBase and your web browser. I can go in and select tracks that I want to load. And if you go here and click, again, I'll do this one more time. So you'll see here there's a Select Track tab in the upper left-hand corner. If I click on Select Track, you will notice that there's a bewildering number of tracks, and it's overwhelming in the beginning. I'm usually overwhelmed when I first look at this, but it turns out JBrowse has nice ways of finding things and categorizing things. So for example, if I'm interested only in looking at RNA sequence data, for example, I can go here and I'll see, oh, there are 848 tracks that are RNA sequence tracks. So I can click on this. This will now show me only the RNA sequence data. I can go down and select a specific experiment if you know it, or I can look for it, right? So I can, for example, just start typing a word like sex or sexual differentiation, and as soon as I type that, it will filter any experiment that had that word in the name of the experiment, or anyway in the description of the experiment, and then I can go in and just select tracks that I'm interested in, and as you click on those, they will start loading in the background here in my Genome Browser, and so I can really configure this as I wish. In addition to selecting by experiment, you can also select by the type of alignment. So for example, that we align things, we align all the unique reads and also all the non-unique reads. And so this is nice to show you when there are regions of gene duplication or just duplication in the genome. You might find a lot of reads mapping to a region, and it's hard to know what part of that region is being expressed because the reads are non-uniquely aligned. I can also select things that are only non-stranded, for example, or if I want to do things that are only on the forward strand or reverse strand from stranded experiments, I can pick those. And so there's lots of things you can configure here. So if I select unique, for example, now it only shows me the unique reads, and I can select, let's say, this one here, which is a unique read coverage. And then when you go back here, everything you've loaded will populate in this section right here. Okay, so quickly, I'm going to jump to the next part, which is the ability to analyze your own data using Galaxy. So we have our own Galaxy implementation. When I click on Analyze My Data, it will take you to this Galaxy interface. If you're not familiar with Galaxy, again, it's not something we develop. We're using a software or tool developed by the Galaxy group, and it's incredibly useful. So I'm not a bioinformatician by training, So having something like this, a web interface where I can do my own RNA sequence analysis and work with the data is really a great thing for me. And so we have this page configured for you with already predefined workflows that allow you to analyze RNA sequence data or do some variant calling. But there are many tools here on the left that allow you to configure and develop your own strategies, your own workflows. And we will have a whole webinar dedicated to this. But I did last night, again, just to show you how straightforward this is, there's a little bit of a learning curve, but last night I just went to the ENA browser. I found an experiment in Anopheles stephensi that looked at male and female mosquitoes, did an RNA sequence experiment on them. I pulled all the data into my Galaxy workspace. I ran one of these workflows here to look at differential expression. The workflow ran in a few hours, and now I have results from differential expression for, now I can look and see which genes are differentially expressed between male and females. But what's really cool is that we have a tool that allows you to export your results back to VectorBase. And then when you export those results back to VectorBase, you can now interrogate your results in an interface that's also hopefully familiar and allows you to integrate the data with other data in VectorBase. So I did that last night. I exported my results here. If I go back to my VectorBase page here and I go to my workspace, you'll see there's a section called My Datasets. And when I click on this, it'll open up all my datasets. This is the one I worked on last night. I'm going to go to this dataset in particular. And you will notice here that there are some options. It tells you the experiment. There's an available search, which I will quickly show you. And also you can view all the results in the Genome Browser. If you click on any of these, it's going to load the Genome Browser view to a particular section. And here's my coverage of the data I analyzed last night. And also I can click on this RNA-seq search. And this shows you the search, just like what Gloria did earlier. You know, she did an RNA-seq query, but this is your private data. This is not the data. This is not loaded in the database for anybody else to see. It's your private data. So I can, for example, say, find me all genes that are upregulated in females compared to males by at least 5-fold. And I can play around with these various parameters here. All of these are described under these question marks, so don't worry about remembering this too much. We'll cover these in more detail in a future webinar, or you can visit one of our recorded webinars. When I run this, it told me that there are 259 genes that met these criteria. I can look at them here. It gives me a custom graph based on my data. So I see these are the 3 female samples, the 3 male samples, and you can see, oh, this is truly upregulated in females. I can go to the gene page itself, and on the gene page itself, in the transcriptomics section, there will be a custom graph that's inserted in that section that shows you the results of your experiment, right? So this would be completely private. So if you go to this gene page on your browser, you will not see this data. So that's a really nice way for you to be able to analyze your own data and work with the data within the database and compare it to other results that are already preloaded in VectorBase. I will stop here because we're running short of time, and we'll cover this in more detail in other webinars. So Gloria, back to you.
Thank you very much, Omar. I'm going to change the presenter back to me. Okay, excellent. So thank you very much, everybody, for attending today's webinar. If you had no time to type your questions or you have not got an answer yet, please contact us via email, contact us, community chat, social media. See you next week we have a release webinar, but the next webinar for this VectorBase series is in 3 weeks, and that will be more in depth about building complex search strategies in VectorBase. Thank you, everybody, so much for attending, and bye-bye. See you later.
Thank you. Bye-bye.